All right. So I guess we've hit 12:01, so we should probably get going. It looks like the number of people joining in is starting to converge, so we will get going. So hello everybody, and welcome to this week's SV Grid webinar series. And this week's talk is very perfectly timed. We have Kirill Nadezhdin and Arthur Neuberger from Alexander Sobolevsky's group in Columbia University, and they are going to talk about structural mechanism of heat-induced opening of temperature-sensitive TRP channels. If you are paying attention to the Nobel Prize news releases, there are some similarities here. So with that, I will turn it over to Arthur and Kirill, and thank you for agreeing to talk today, and thanks everybody for joining.
Thank you so much, Pete. Let me just get my presentation up and running. Perfect. Thank you so much for this invitation and this possibility to present our most recent research here. My name is Arthur. I'm a Walter Benjamin Fellow in the Sobolevsky Lab here at Columbia University. And today, together with my colleague, Kirill Nadezhdin, we are going to present to you our most recent research on the structural mechanism of heat-induced opening of a temperature-sensitive TRP channel. And as Pete already said, the timing that we've chosen for this presentation couldn't be more perfect. It's been only yesterday that the Nobel Prize in Physiology or Medicine was awarded to David Julius and Ardem Patapatian for the discovery of receptors that enable us to feel temperature and perceive touch. So also from our side, our sincere congratulations to both recipients. TRP channels, or transient receptor potential channels, are cation gating membrane proteins that are categorized into 7 different families and are activated by a variety of both physical and chemical stimuli, including, but certainly not limited to, temperature, touch, pH, and a long list of both synthetic as well as natural compounds, some of which are displayed here, in response to which these channels open up, are activated, allow for ion flux into the cell, and with that trigger a variety of different signaling cascades, in response to which we are able to sense our environment and make rational decisions about it. And we are particularly interested in those members that are activated and respond to different temperatures directly. So they're thermal TRP channels. And I don't need to mention how quintessentially important it is for us to be able to feel and perceive temperature. We grow up learning to not touch fire. Maybe you do it once and never again. We know do not sip on boiling hot coffee or tea or hot beverages. We know we need to feel cold in order to preserve ourselves and seek shelter when we need to. And all of these mechanisms help us to preserve ourselves and preserve our body. And although we perceive temperature really in a spectrum, this process actually this job is shared by a variety of different TRP channels, each and single one of them operating in their own respective range, temperature range, and being activated in their own respective temperature range. And we generally distinguish between cold sensors and heat sensors. And one such heat sensor in our body is the vanilloid member 3 here, TRPV3, which will be the subject of our research here that we are presenting. TRPV3 is present throughout our body, but it is especially well expressed in our skin and skin keratinocyte cells. It fulfills a whole range of basic important physiologies in our body, and being especially present in the skin, it contributes to a healthy skin barrier, to hair growth, and the ability to sense temperature through our skin. So these days, if you go out to the balcony and check what to wear in the morning before going to work, it's basically TRPV3 that signals you, hang on, you probably have to wear a jacket in order to not freeze. It also contributes through mutations and through especially gain of function mutations to a variety of diseases, including some really horrible skin diseases such as onset syndrome displayed here. So there's a whole bunch of, there's a whole pipeline of research trying to investigate the molecular mechanism of activation or inhibition of TRPV3 through different small molecules, some of which are derived from natural compounds, from nature-inspired. And this nature-inspired drug design is really important to target some of these diseases. However, for the research that we're presenting here today, you are not looking into the agonistic activation of TRPV3, but we set ourselves the goal to solve the first heat-induced thermally activated structure of a wild-type TRPV channel, and here in the case of TRPV3. One of the key aspects to be successful in this journey for us was to early on recognize that we have to reconstitute our protein that we solubilize with regular detergents, pull out of the cell with regular detergents into nanodiscs. And for those of you who are familiar with nanodiscs, working in nanodiscs, know that these are basically lipid donuts that are held together by a scaffold into which the protein can reconstitute, and we can therefore shield its transmembrane domain against the aqueous buffer surrounding it. Now, the deliberate approach to work in nanodiscs is based on three important observations that we made early on into the study. One of these observations is that if we apply heating protocols to our protein that is solubilized in detergent only, we can see that through the application of heat, by heating it to temperatures above 40 degrees, and this is what we need to activate this heat sensor, is that we see that the protein actually degrades, it aggregates, it's really burned through the application of temperature. If we reconstitute it in nanodiscs, this protein is much more stable and it survives the heating that we apply in order to open it up. So it's quintessential for us to keep the protein healthy when we apply heating protocols to open it up. Second important point, and this really goes along with a growing body of literature that suggests that the local lipid environment really allosterically contributes to the activation of many membrane proteins, including thermally activated TRP channels. So, we do see lipids sticking to the transmembrane domain when we purify the protein out, solubilize it out using detergent. However, we can't rule out that once we start to heat the protein, that these lipids are potentially need to be exchanged through lipids in the local lipid environment. And in order to sort of supply the physical and chemical properties that are likely similar to the natural environment in the transmembrane, we decided to work in nanodiscs. And the third reason is really more of a practical, pragmatic nature for the sake of doing good cryo-EM. Once we reconstitute the protein in nanodiscs, it really starts to behave more like a soluble protein. We can suddenly freeze grids with very thin ice and can collect in very thin ice. We can see different orientations, and that helps us to solve a really high resolution structures of it. I'm going to show you here one such example micrograph, and you have to take my word for it. This is really a representative micrograph of TRPV3. We see beautiful monodispersed particles. It's not even the prettiest micrograph that we have, but you can already appreciate that we can see the particles in different orientations. And if we try to reconstitute sort of 2D class averages, calculate 2D class averages on these particles, we can see particles in different orientations, and we can already start to see secondary structural elements. Now, if we process this data all the way to a final map, we've been recently able to solve a stunning sub-2 angstrom map of the full-length wild-type TRPV3 channel reconstituted in the nanodiscs. And this is a magnificent map. We see beautiful water molecules. We have beautiful side chain densities that allow for very precise modeling of a structure into these densities. And this structure you can see here rotating on the right, it's typical for TRPV family channels. It's a tetramer that forms the ion channel conductance pathway in between these four subunits across the membrane. And what you can also see are these chunky anchoring repeat domains, and that altogether we like to call the skirt. And I'll show you in the next few slides how the skirt also contributes to the thermal activation mechanism of TRPV3. Now, potentially even more important than having a nice specimen that gives us beautiful cryo maps and allows for precise modeling of the structure is the fact that our protein that we prep is also active. So what we did here was to ship actually, so the same protein that was used for this structural investigation, in fact, not just from the same prep, from the same Eppendorf tube, was shipped to our collaborators in Illinois to the Zakarian lab that do magnificent single channel bilayer recordings. And so what they do here is they take purified protein, reconstitute it in a bilayer, and probe it on channel conductance. They measure single channel conductance, or single protein conductance rate. And normally these recordings are done by the addition of various agonists to activate the channel. In this particular case, however, we have a heat sensor. So heat alone, temperature alone, should be able to open this channel without the application of any additional agonists. And this is exactly what Ella and her colleagues did here. The purified channel, a single channel here in the chamber, where we ramp up the temperature to 36 degrees and above, and we start to see channel openings. And once we reach temperatures closer to 42 degrees, we see significant opening events of the channel. So this is very good news for us. Because the protein that we prep is also active, and we really need active protein in order to solve heat-activated open structure of it. So these are representative traces, as you can see here below, of what Ella and her colleagues have measured. And you can see that the protein behaves exactly as it should as a heat sensor. And it's very, it's overlapping well with the data that has been physiologically shown in whole cells. The protein stays completely closed at 25 degrees. And once we start to heat the chamber, we can see opening events at 36 degrees, and the channel really reaches maximum open probability and channel conductance rate at 42 degrees. Another important observation that we made along the way, and this is really a work from Victoria Vlachova's lab, is that if we now don't apply just a regular constant application of temperature, but once we start to cycle temperature between heating and cooling, so say 30 seconds at 25 degrees, followed by 30 seconds at 42, back to 25, back to 42, and if we do it for long enough, for 10, 15, or 20 cycles, what we can achieve is the maximum open probability and maximum conductance of our specimen. Now, think about it. This is very important for us, because what we eventually want to do is to plunge freeze our sample in liquid ethane, and we want to have a significant proportion of our particles to be in an open state in order to have enough particles that would allow us to reconstitute, to process out a nice density map for a putatively open state. Another important observation that we made at this point was that TRPV3 has a slight delay in its closing when we drop the temperature back to room temperature, back to 25. And this delay, which is a couple of milliseconds, really gave us hope, because if you think about the plunge freezing process, for those of you who are doing cryo-EM, you will be aware of how the arm of the VitroBot group leaves the chamber, plunge freezes with the tweezers and the grid attached into liquid ethane. We know from literature that liquid ethane freezing can take quite a bit of time. We're talking about milliseconds, depending on the literature that you refer to. It certainly varies with the actual temperature physically achieved on that day when the liquid ethane was prepared. It depends certainly on the quality of the ethane, of contaminations, etc., etc. So there's a chance for us to actually lose the protein at that point and actually get 100% closed particles. But with this little delay, and in combination with this procedure that we call thermal sensitization, there's a chance to capture at least a fraction of protein in an open state, so that it is frozen quick enough before it starts to completely close up again due to the exposure to the cold temperature. And obviously, liquid ethane is very cold. So what followed was really weeks and months of optimizing this heating protocol, the cycling protocol. We tried a different amount of cycles of steps. We varied temperature. We tried different time points, exposures, until we came up with what we thought the ideal, we like to call it the SPA protocol for thermal therapies. And the way we would do these experiments is literally just to take our thermocycle, PCR machine, down to the VitroBot room, place it right next to the VitroBot, cycle the protein through these repeated heating and cooling cycles, so sensitize it to achieve maximum opening, the maximum amount of particles of purified protein to be open at that point. And when in the 20th or so cycle, before the temperatures drop to 25 degrees room temperature, we would have the equivalent of these 25 degrees to actually pipette the protein out and transfer it casually and pipette it onto a grid that is attached to tweezers already inside an equilibrated VitroBot chamber. And that VitroBot chamber is set again to 42 degrees, 100% humidity. So kind of simulating yet another heating cycle where the protein in this environment that really resembles like a Turkish hammam, it could relax one more time, open up one more time before it is then rapidly plunge frozen liquid ethane. And at that moment, we thought about it. we had a chance, due to this sensitization and this delay, to capture at least a fraction of particles in an open state. And we are really hoping that the freezing is quick enough and that the protein stays open long enough to see at least something going on. And when we made our first grids and collected our first preliminary data set and processed it out, we saw the following, what you're going to see in this video, and we are really pleased and happy to see that, because what we saw in the particle stack that we extracted, this significant movement of the anchor and repeat domains of the skirt going up and down, as well as if you look from the intracellular side, the bottom view, you can see the significant rotation of the skirt that goes along to what looks to us at that moment like an opening and closing event of the channel. So there's considerable heterogeneity in our sample, and that really was, a real eureka moment for us. We knew at that moment, we didn't know whether it's like a fully open state, but at least we knew that our protocol seemed to have worked, and we have really pushed this protein into a range of different conformations. So what followed obviously was a real tour de brute force. We collected an enormous amount of data, and one of the key factors here was really to very carefully clean the data, classify particles out until they can't be classified anymore. That gave us pure, clean conformations. So let me guide you through what we've seen. And we started off, as I said, working in nanodiscs. We started off with MSP2 and 2 type of nanodiscs. I'm sure most of you will be familiar with that type. And the first thing we did was to collect a 4 degrees control, because at 4 degrees, we expect our protein to be completely closed. It's a heat sensor, it shouldn't open up. Anyway, we collected this data and built a structure. 100% of our particles in this data set actually aligned to this one single conformation. And then if we use Kirill to calculate the profile of the channel, we can see several tight constrictions, one in the selectivity filter and one further down formed by these asparagine and isoleucine residues that form this really tight constriction in the lower part of the channel. At that moment in time, we realized what we are facing with this data, with this heterogeneity, is probably a different set of conformations. And we didn't want to speculate about whether a channel is open, half open, semi-open, fully open. And we really wanted to have an independent assessment of whether the conformation that we have is in an open or closed state or anything in between. So we decided to collaborate with Roman Efremov and his colleagues in Moscow, who do beautiful MD simulation work on trip channels. They're very specialized on that. And they confirmed independently that both water molecules here depicted in blue, as well as ions here depicted in red, cannot travel through this channel. It is completely closed up. And this is good news for us because it's supposed to be closed. It's a heat sensor after all. Now, when we started to apply our cycling protocols and we processed the data, we could now suddenly squeeze out 2 distinct conformations. And the new conformation that was added up here, as you can appreciate, has already some structural changes. And what we can see inter alia, and this is something that should remind you of this little video that you've seen, is a lifting of these anchoring repeataments, a lifting of the skirt that comes along with a very interesting and intriguing rearrangement of the N and C terminal tails. And I'm going to talk about it in a moment, what that signifies. Now, when we calculate the channel radius, the channel profile, we can see already that in this next different conformation, we have a slight widening of the selectivity filter as well as of this constriction side. Once again, we didn't want to make any speculations, and we consulted Efremov and colleagues to do the MD work. And interestingly, they confirmed that we have 2 distinct subpopulations in the sample. We have a fully closed state, putatively because some of the particles never opened in the first place for whatever reason, or because some of the particles actually could manage to close due to exposure of the cold temperature prior to be completely frozen in ice. The second conformation, however, is very interesting because it looks like a semi-open state, and we label it sensitized state, where water and ions can sort of enter the channel, but they can't quite get through it. They're still hindered sterically and electrostatically from complete passage. So it's a semi-open state. At that point in time, obviously, we had 2 major directions that we could follow. We could either apply more brute force, more heat, longer cycles. But we also realized that at this point, we had optimized it. And if we apply more heat, high temperatures, more cycles, we'd probably lose the protein, because some of it, even though it reconstituted nanodiscs, won't be stable for a long time. Instead, we thought more carefully about the type of nanodiscs we're using for reconstitution. And we decided to actually try different ones, because we had a suspicion that, maybe it's the nanodisk that restricts its movement or creates a not ideal local lipid environment that restricts a full opening of the channel. So instead, we tried this different type of nanodisk. Some of you might be familiar with it. It's been pioneered by the Wagner lab in Harvard. It's the circularized CNW nanodisk. And we used CNW9 because it was the perfect fit size-wise for our transmembrane domain in the tetramer. And again, we started off by just collecting the 4 degrees control to make sure the protein looks good and hopefully close at 4 degrees in this type of nanodiscs. And we got indeed a closed state that we confirmed again independently by Efremov to be fully closed. Now, once we start to apply the same heating protocol that we had applied to MSP2 and 2, we again could squeeze out 2 distinct classes of particles in 2 different states. And this second state that we now achieved looks familiar, right? It looks like the sensitized state. We have a lifting of the anchor and repeat domains, we have a rotation here, and we have a rearrangement of the tails. Now, the most important question at this point is it now eventually open or not? Now, when we look at the channel profiles, what we see compared to the other conformation, which again is a closed state as we expected, we can see a significant widening here now in the selectivity filter, as well as further down in the site where we had the constriction previously in the sensitized state. We were hopeful at this point, but we really wanted to make sure that it is a fully open channel. Once again, we consulted Efremov and colleagues to do the MD work, and eureka, it is a fully open channel. They could show that ions pass freely through that channel. It is a completely open conformation, which was exactly what we wanted to achieve. So in a nutshell, what we've achieved is the first thermally activated open state conformation of a wild type TRP channel without the application of any agonists, without any mutations or truncations. It's pure wild type activated by heat and heat only. What we've also achieved through our experiments working in a different type of nanodisc is a so-called sensitized state or semi-open state. Now, you might wonder whether this state is real, whether it's a real transitioning state towards an open state. And actually, we believe it is real, and it was really an act of serendipity that we achieved that in that particular type of nanodiscs. Because structurally it resembles, it's very close to a lot of structures that had previously been solved in the presence of agonists through mutations, where people tried to push TRPV3 into an open state. So it resembles some of these structures, and we believe it is a real transitioning state towards an open state. We have also solved 4 independent structures of TRPV3 in the closed state, 2 in 2 different types of nanodiscs, and obviously 2 in different types of temperatures. And just to emphasize, obviously 100% of our particles in the 4 degree control were in a closed state, and only a fraction of particles in our heat protocols was closed. And again, we can speculate about whether this is due to the particles never opening up in the first place, or whether they managed to close prior to being completely frozen in ice for eternity. So let's sum this up. Through the application of this thermocycling and heat sensitization, we achieved 3 different conformation states: a closed, a sensitized, and an open state of TRPV3 wild type. And we confirmed that these states are indeed closed, semi-open, and fully open. Now let's look at some of these structural alterations that we see between these states. And the obvious thing to do here is to compare the closed state to the sensitized state, as well as the closed state to the open state. And in both cases, in the sensitized and the open state compared to the closed state, we have this significant rotation of the entire skirt domain that comes along with lifting of the anchor and repeat domains. And that goes along with a structural rearrangement of the N and C terminal tails. Now let's look at this more carefully. What does it mean? What is going on here? So what we see is that this proximal domain with these beta sheets from one protomer and the anchor and repeat domain of the neighboring protomer form a little groove, a little cavity. And this cavity is in the closed state occupied by the C-terminal tail of the upper protomer. So what this tail does here, really, it interacts with different residues in both protomers and acts like a little glue, like a little loose log to make sure that these protomers stick together, and it stabilizes this closed state. Now, once we start to apply heat, we have the significant rotation of the skirt, we have the lifting of the skirt, and we have a rearrangement of the tails. And what is happening is that this C-terminal tail now curls up into a little helix and makes room for something else to fill this cavity, this groove here. And what is filled is with the, what fills up this groove is the N-terminus from this lower protomer that now comes in and occupies that cavity. And we don't see this N-terminal in the closed state because we expect it to be, floppy, and only when we apply heat and push for a structural rearrangement, for a conformation shift towards an open or sensitized state. And once this space is freed up, the tail comes in and occupies this space. It again stabilizes the interaction like a little glue here between these two protomers. And computationally, again, our colleagues Efremov et al. Have confirmed that actually both states are energetically stable, and that in one case, this stability is facilitated by this C-terminal tail here and the gluing between these protomers, and in the open or sensitized state, it is the N-terminus that facilitates that junction here. Now, we looked at what is going on now structurally in the shift from a closed to open or sensitized state. And one thing that we haven't talked about yet is lipids. And when we compare the closed to the open state, and because really due to the fact that we have such a high quality map in both cases for the closed and open state, we can really compare lipids against each other. And in both cases, we see beautiful lipid densities here highlighted in blue, and we compare them against each other, every single lipid in one map against the other in the other map. And we see that they're almost perfectly identical with one prominent difference. And for those of you working on TRP channels might already suspect where the difference comes from. And yes, indeed, it is a lipid in the vanilloid pocket, a key allosteric site, where we have a beautiful lipid density here highlighted in red sitting in the closed state. And this lipid is gone in the open state. It's popped out, destabilized. We don't see any evidence for density in this pocket. So we have a suspicion that this popping out of this lipid through the application of heat, Brown motion, really maybe initiates something like a conformational shift towards another stable conformation, an open or sensitized state. Now, at that point, we also wanted to confirm which lipid it could be. So we once again consulted Efremov and colleagues to do some MD simulation work on it to see whether there's any preference for a specific lipid type to occupy this pocket. And interestingly, the simulations suggested that although there is some statistical variability, there's no single candidate to occupy the space. Different lipids can occupy the space in the closed state and stabilize the closed state. And once we start to heat the specimen, this lipid, whatever lipid it is, at that moment is popped out. At this moment, I'm going to stop sharing and hand over to my colleague Kirill, who's going to guide you through different mechanisms of activation. Kirill, the stage is yours.
Thank you very much, Arthur. Let me share my screen. I hope you all can see my slides now.
Yep, looks good.
Okay, so my name is Kirill Nadezhdin, and I work as a postdoctoral research scientist in Sobolevsky lab, and I want to go on with this research. So the term vanilloid pocket or vanilloid lipid came from the name of the binding site of capsaicin to TRP1. So in 2016, David Julius and Yifan Cheng proposed that heat-dependent displacement of vanilloid lipid plays a role in activation of TRP1. But all structural work on TRP1 channels at that time was done only on truncated constructs. So no structure of full-length TRP1 was available. So we decided to fill this gap. So this work was done in collaboration with our colleagues from Yale University, Elena Grachova and Slava Grantsev labs. And in this work, we used cryo-EM and obtained structures of full-length TRP1 channel from a kit animal, a 13-line ground squirrel. You can see the picture of it. These animals, these hibernating animals have evolved in such way so they now can tolerate cold and high and low temperatures for a long period of time, which allowed them to live in areas that are too hot or too cold for our other species. And among other things, they developed tolerance to heat by modifying TRP1. So in our work, we saw the structures of TRP1 in 3 different states: in apo state without any ligand, and in complex with resinophyllotoxin, the toxin from plants, and capsaicin from hot chili peppers. And I have to mention that RTX and capsaicin are strong activators of TRP1. So here you can see our structure of full-length squirrel TRP1. The structure is similar to other TRP channels, similar to TRP3. And in comparison with previously published data, our structures show conformations of the cap domain that is shown in blue and C-terminus, C-terminal domain that is shown in pink. Both these domains were previously truncated by other groups. C-terminus connects membrane proximal domain of one subunit to ankyrin repeats of other subunit. In other words, C-terminus acts as a glue between 2 subunits. And we observed some movements of N and C termini in TRP1 during heat activation. So similarly, both N and C termini may contribute to temperatures gating of TRP1. So in fact, supporting this idea, the mutation of asparagine to serine converts weakly temperature-sensitive TRP1, squirrel TRP1, into highly temperature-sensitive channel. So this asparagine is located at the very end of ankyrin repeat 1, in close contact with the ankyrin repeat 2 and close to C-terminus. And it was shown that ankyrin repeats 1 to 4 do not contain heat sensor by themselves, but this region can allosterically influence actual temperature sensors, probably C-terminus. So here we compare our structure with previously published structure of red TRP1. As you can see, they both look the same, both globally and the channel pore. But now we can see cap domain and C-terminus. Recently, actually, 2 papers have been published with full-length structures of red TRP1, one by Lee Lab from Duke University and another one by Kai-Hua Zhang, David Julius, and Yifan Cheng from UCSF. But again, their structures in apo state turned out to be the same as ours, but we clearly see cap domain and they don't. So let's talk more about the organization of cap domain. We see in our structures that 4 subunits form a turret above the channel pore, and we see the strong density in the middle of the cap, which we think is a chloride ion. And it represents a barrier for cations. But on the other hand, there are 4 side portals that lead to the pore. These portals contain many negatively charged residues that are shown in red on the channel surface. And these negatively charges create a favorable environment for conduction of positively charged ions through these portals. So why cap domain is so important? When we truncated cap domain and recorded spontaneous openings from single channels, we observed that open probabilities of these 2 constructs are not the same. So with the help of our colleagues from Yale University, we did functional studies, and the amplitude histograms revealed that deletion of cap domain produced significant increase in single channel conductance from 125 to 174 picosiemens. And compared to wild type, the mutant showed flickering behavior, probably we think due to significantly decreased mean open dwell time. And moreover, when we truncated this cap domain, the truncated constructs were not activated by heat, as you can see on this slide. So when we applied temperature ramps, channel didn't open. At the same time, the channel was still responsive to low pH or capsaicin. So can TRP be heat sensor? Previously, Tin lab from State University of New York at Buffalo showed that cap domain is not essential for temperature sensing. On the other hand, results from the G. Zhang lab from UC Davis argued that cap domain is indeed a part of temperature activation pathway. So what else did we find in our study? The quality of our data allowed us to see many lipid densities that interact with the channel that are shown in blue. One specific regulatory lipid, the famous vanilloid or pocket site, and most probably it is occupied by the phosphodiazole. And the difference between APO and ligand-bound structures are most obvious when we compare channel pores. So this lipid can be replaced by agonists, either RTX or capsaicin. So while the APO and RTX states have similar pore organization, the pore in capsaicin-bound state have completely different organization and conformation. So we see pi to alpha transitions in S6 helix that forms of the pore. That has never been observed for TRP1 before, but it was later recently confirmed by the study of red TRP1 by David Julius and Yifan Chen groups. The channel's lower gate is completely closed in all states that we solved. And it is sealed by isoleucine 681 in APO and RTX state and by methionine 684 in capsaicin-bound state. It is interesting. Actually, RTX and capsaicin are strong agonists for TRP1, and they both should open the channel. But capsaicin-bound structure and RTX-bound structure are both closed. It's kind of a puzzle for us. But recently, the study from Lee lab from Duke University has shown that indeed, TRP1 could be opened in cryo-EM by applying both heating and capsaicin. So let's discuss how TRP channels actually sense the heat. Many studies were done to find the molecular temperature sensor for TRP channels. And one possible candidate is shown on this slide. Here you can see TRP1 structure from bottom side and top view. And one subunit is colored in blue, and the region of interest, the possible temperature sensor, is colored in red. So Feng Qin showed that N-terminal region, membrane proximal domain, is crucial for temperature sensing. They also speculated that ankyrin repeat 5 and 6 may play a role in temperature sensing, most probably allosterically due to interaction with membrane proximal domain. And this is the work from Kenton Schwartz lab. They showed that heat activation is intrinsic to pore domain. They used structure-based engineering between heat-activated TRP1 and the shaker potassium channel. So they showed that insertion of TRP1 pore domain into shaker resulted in functional channels that can be activated by toxins that are selected to TRP1 and by noxious heat. And also NMR studies of an isolated S1, S4 bundle or voltage sensor-like domain show that S1, S4 also significantly contributes to thermosensing. So, and this study, I believe this is my favorite. This work was done by the group of Ramon Latorre at Austral University of Chile. And this studied the role of C-terminal domains of TRP1 and TRPM8, which is the cold sensor, were questioned. They swapped C-terminal domains between TRP-B1 and TRP-M8. And it turned out that TRP-1 with the tail from TRP-M8 became cold sensor. At the same time, TRP-M8 with the tail of TRP-1 became a heat sensor. So they called that hot sensing cold receptor. Beautiful work that shows that heat or cold sensation are determined by C-terminus. So, taking all together, it seems that almost entire sequence can be named as a thermosensor. In fact, there is a theory that proposes that the entire protein can be a temperature sensor. In other words, many residues sense changes in ambient temperature, and those residues are distributed across the protein structure. And Clapham and Miller proposed that the change in heat capacity between closed and open states can explain the temperature dependence of activation. So exposure of a few nonpolar residues to solvent after heating is enough to explain high changes in entropy and enthalpy. And according to this principle, the group of Baron Chanda from University of Wisconsin-Madison converted voltage-gated channel shaker into a heat sensor by mutating some residues. So that was all about TRP-1, but what is known about TRP-3? Here in red are shown some residues that are proposed to be involved somehow in temperature sensation on TRP-3. Most of these residues are located close to the extracellular loops, but some are also located close to TRP helix and S2S3 loop. Luckily, we were able to open wild-type TRP-1 by heat, so let's go back and try to analyze the differences between the closed and temperature-activated open states of TRP-3. So, first of all, we see changes in secondary structure during heating between closed and sensitized states. These changes include S2S3 linker, ankyrin repeat 5, and N and C termini. No such changes occurred during transition from sensitized to open state. So we believe and we propose that the first step is highly temperature dependent, and the second step is weakly temperature dependent. And these changes in local secondary structure translate into global movements of ankyrin repeats, swapping of N and C termini, and in the end, dilation of the pore and opening of the channel. So these huge global translational movements we call conformational wave. And this is a short video that illustrates what I've just said. I hope it will work. So you see the closed to sensitized transition with the dramatic changes in the skirt, and it goes up and with respect to transmembrane domain. And sensitized to open transition is really localized to the gate area. This is very important because during this transition, the channel actually opens. So, and here it all goes backwards. And also there is a bottom view. You can see the formation. Here you can see the formation of alpha helix in C terminus and overall rotation of the skirt. And now you see the opening of the ion channel. This is very localized. And reverse transition, rotation of the skirt, you will see it. And exchange between N and C termini. So to summarize it, altogether, we have solved, we have 3 different states: closed, sensitized, and open states. And we think that closed to sensitized state is highly temperature dependent, and sensitized to open state is weakly temperature dependent. The closed to sensitized state, the closed to sensitized transition occurs, we think maybe the trigger is the lipid, and the changes in secondary structure of S2S3 linker, and all this propagates as a conformational wave to the linker domains, membrane proximal domains, the skirt, swapping of NC termini, and formation of alpha helix. And after that, and at this stage, the channel is still sensitized, but it is still closed. But at the last step, we see local movements of S6 helices and opening of the channel. So let's go to the conclusions. We captured full-length wild-type TRP1, TRP3 channel in 3 different states: closed, sensitized, and heat-activated open. The heat-induced conformations include changes in secondary structure of linkers and swapping of NNC termini. And heat-induced transformations include dissociation of lipid from this vanilloid site. And we think that it plays an active role in temperature gating. And all these changes in conformations of linker, termini, and lipids are all linked together to other rearrangements in other parts of the protein. And all together, we name it heat-induced conformational wave. And on top of that, we saw the structures of full-length wild-type TRP1 channel, and we clearly see a cap domain and C-terminal domain. And we showed that both termini and cap domain are critical determinants for TRP1 function. And I want to thank, in the end, all members of our lab, and particularly Arthur, with whom we did all this structural work together. We are working as a team. I want to thank our collaborators from the Yale University, with whom we did our work on TRP1, and international collaborations with these labs. So we were able to capture and explain the temperature sensation TRP3 channel cryo-EM facilities that helped us to collect beautiful data sets that allowed us to solve the structure of TRP3 to the resolution better than 2 angstrom, and all the funding agencies. And I also want to thank you all. And if you want to know more about our work, please ask us questions, and you can also go to our papers. So, thank you.
Thank you very much. That was, both of you, that was a good tag team effort at explaining heat sensitive channels. So thank you. That was very interesting. And yes, we have somebody else who did the clapping emoticon, which I don't know. It doesn't feel the same as normal clapping, but you know, it's better than nothing. And yeah, so for folks who, if you have questions, you can use the raise hand reaction, and we'll unmute you. I guess one question that occurred to me as you were talking about the nanodiscs, but may also be related to the vanadidium pocket, is are there phase transitions in the lipids in the bilayer at the temperature ranges that overlap with the TRP V channels? And is that something that might possibly play a role?
It might be, yeah. At the moment, we don't see this in the simulations. I guess these simulations would be very long and very complicated to run to see how different lipids pop in and out. But there's some data from other TRP channels that we are processing and working with, where it is suggested that actually lipids externally come in and exchange some of that lipid environment that's present. It would be really fascinating to simulate that and see maybe in some more advanced simulations how, you know, this lipid environment really interacts with this transmembrane domain. Maybe some of the lipids pop out and others come in. It's an interesting thought, but yeah, it's hard to do. I mean, I guess you could try to go with labeling and design experiments functionally to show how certain lipids leave and come in, but it's an interesting thought, and it's definitely something worthwhile pursuing.
Yeah, well, I'm also reminded of the, you know, one of one of the researchers I'm familiar with had done just pure bilayer studies with, lipids that will be in an ordered phase and then shift to a disordered liquid phase. And is that related to membrane rafts? And so it seems like I don't have a good experimental avenue here, but it seems like there might be some overlap.
Yes, absolutely. And I wanted this actually lipids is kind of controversial topic, and some labs show that lipids do play a role, like for example, specific lipids like cholesterol, and other groups show they don't play a role. And again, these phase transitions in lipid, it's also kind of interesting because this phase transition should occur at like one particular point, like one specific temperature, but we have like a lot of temperature sensitive channels, and they sense temperature like from 10 degrees to 50 degrees. And it's hard to imagine how those phase transition like all play together. So maybe for some channels, it may play a role, maybe for others not. So yeah, it's kind of very interesting topic. We're also thinking about that, but still we don't have the answer.
That's why we do research, right? Like I guess just to shift to a more, I guess, practically oriented follow-up on that, for the two different types of nanodiscs where you were seeing for TRPV3, the sensitized conformation after the heat protocol and then the open conformation, what do you think the difference was between the two types of nanodiscs?
We have a suspicion that there might be, well, we don't have proof of that, but there might be both a steric component to that, steric, constriction of movement. But it could also be due, and this is really referring to some MD simulation work that's been done in nanodiscs with lipid environment purely, where we see that in different lipid nanodiscs, we have a different organization of lipids inside those. And it might be both. It might be a steric restriction that prevented us from opening up fully, but it might also be a combination of certain, lipids being organized maybe in a non-ideal fashion inside this nanodisc. We again have no proof of that. It would be interesting. In an ideal case, we would see every single lipid at high resolution in the surrounding environment, and we could see really what is going on. Whether we can see different patches of specific lipid type being in the local environment. Unfortunately, we cannot see that, but we also cannot outrule that this is indeed happening in a less favorable way in one nanodisc against the other. But size also matters. And that could also be a contributing factor for sure.
So for other experimenters who were working on proteins in nanodiscs, it sounds like sometimes you may have to try different variants, and it's empirical.
It absolutely is. Yeah.
That sounds oddly like crystallization. That's where I've got my background. Sure. And again, just to remind folks in the audience, if you have questions, please use the raise your hand reaction. Otherwise, because I don't want to be monopolizing questions. So we do have one from Jevon GC. So I will see if I can work the interface and figure out how to get you to unmute. There we go.
Yeah, thank you. It's a great talk. I have one question, so the lipid composition is different in between the normal cell and disease cell. So how do you think the structure will adapt in this kind of disease cell?
So yeah, this is kind of a great question, and we don't have any answer, like any good answer we don't have, but we have some answers to that. So actually, when we tried, at least for TRP3, we tried different type of lipids. And we also used mixture of lipids, for example, soybean lipid polar extract. And in case of TRP3, we didn't see like much difference in the structures. But there is a lot of work on TRP1. And it showed that lipid composition for TRP1 is like very critical. For example, negatively charged lipids, negatively charged polar heads lipids, they have inhibitory effect on TRP1, and sometimes they have activating effect on TRP1. So yeah, lipid composition is like very critical. for some protein, but maybe not so critical for TRP3. But yeah, I can be wrong. Again, this is just my thoughts on that.
Yeah, because the only reason is so there's a huge difference in the composition between, like for example, the cancer cell and the normal cell. You have a drastically different lipid composition. So my just curiosity was how does these receptors adapt? Because if you want to treat some disease, and then you want to design some small molecules or any kind of molecules to target these receptors, I think we have to look at the environment around these receptors. So I believe this, in addition to these structural changes, the lipid environment close to this receptor will be crucial to design some molecules. Yeah, but you're right. It's very difficult to decompose the effect of lipid composition and the receptors. Thank you.
Maybe in addition to that, there are certain methods where, using these small dip bulbs, where you can potentially try to pull out the protein purified out of its natural environment together with localized lipids. So that's an approach that would be interesting. It's a rather difficult prep. It doesn't work that well in most cases, but that could be an approach certainly where instead of purifying protein out and losing most of the lipids on the way and then exchanging through a non-disc environment with an addition of artificial lipids or even extracted lipids, you could certainly try to attempt that. But yeah, it's one possible approach.
I guess one other question that I've got, if there are no more from the audience, and again, this is kind of a informed speculation / naive question, but you'd mentioned pathology is caused by mutations in TRPVs. Is that possible indications that these channels are doing something in addition to acting as temperature sensors or other types of sensors, or is that just a developmental side effect? I guess it's potentially likely to be, rather than is or isn't, because it's open-ended question.
I mean, certainly these channels fulfill a variety of other roles for physiology. It's not just temperature sensing. B3 contributes to maintenance of a healthy skin. It contributes through various signaling pathways to normal hair growth, and all of these. So they do have a variety of roles beyond just the sensation and temperature, feeling, sensing. So definitely, yeah, more and more studies are coming out, and we learn more and more about the physiological roles in the body beyond the sensing of temperature. And a lot of these TRP channels are not thermosensors in the first place. So they're means to import calcium into the cell. They're involved in all sorts of signaling. So yeah, V3 is an interesting example because it can do both. It allows us to feel heat. I'm wearing a t-shirt now because I feel warm. TRPV3 tells me that I don't need to wear a jacket. But on top of that, the skin that we have and the maintenance of it is also part of what TRPV3 does and contributes to. So definitely, yeah, various functions in the human body.
Yeah. Thank you. Again, thank you both for presenting, and thank you everybody for attending. I do have one final non-scientific question, which is, if our labs or capsaicin is a reagent, do they tend to eat more spicy food or less spicy food, or is there no correlation? Or we have to do an experiment to find out. So.
Good question. No.
Yeah.
We actually, in celebration of the Nobel Prize, Kirill yesterday put some enormous amount of chili peppers on his pizza for lunch. So, in celebration of capsaicin, we challenged our receptors yesterday.
Some people enjoy doing that.
Absolutely.
Yeah.
Great. Well, thank you again. Those were both excellent talks.
Yeah.
Thank you all. Have a great day.
Thank you.